Swayam Prabha. Digital India. Educated India.
So, good morning everyone. So although he has talked about my research interests, I'm not going to talk anything about my research today because it will be Greek and Latin for some of you at least. So let's start something very basic to look at so that it will be useful for you in a long run as well. Now I have a picture here on my screen, right? So basically there are 4 different uncorrelated examples I have. One is basically trying to eradicate the infectious insects, and the other is to increase food production, the other is to develop organ resources, and the other is to rebuild your extinct animals. So these 4 are completely unrelated phenomena, or not phenomena, these are unrelated problems that you are trying to understand, right? So can you think of any one solution that would address all the 4 of those? DNA. You have seen the slides before? No. Okay, so why do you think DNA will address that? How is DNA related to all this? So I have given 4 very different problems here, right? So, okay. Now one is eradicating infectious insects, and then I have an increased food production, and then I have a developing organ resources, and then rebuilding extinct animals. Now, if you look at all these, what we are addressing is something related to your life, right? So all these have some form of life in them, right? Although this is an extinct animal, it had life at some point of time, right? So now to address any of these things, right? If all these have to be addressed in one specific manner, we have to address the basic unit of life. And what is the basic unit of life? Cell. And what does cell consist of? Okay. Oh. Can you try that? So the cell, the basic unit, I think you all have to concentrate back here. And so if the basic unit of life is cell, but what cell consists of is DNA, right? So any life as such, if you have to modify something in the life, or if you have to do anything with the life, in the sense you have to tweak some changes in the life, what you have to do is you have to change how you have to change the basic unit of your life, that is your DNA. It's not working? Can I use the... Okay, so while they sort it out, let's see. So today I am going to talk about something called as, I don't know if you have looked at the topic of my title, it is called as molecular scissors to understand, or molecular scissors to edit the code of life. That is the title of today's topic, okay. So how many of you are from class 11 and how many of you from class 12, or how many of you are going to class 12? All of you, right? Okay. So do you know what are molecular scissors? You don't know molecular scissors. I think it's an important topic in class 12. So if you have not, so probably you'll learn about that. So today we are going to understand what are called as molecular scissors to edit code of life. Now, what is the code of life? DNA. Why do you think DNA is the code of life? Okay. So basically, as I told you, so in order to do any of these changes, you actually have to edit the genome. Okay. We'll come back to it in a moment, what genome is. But genome is nothing but your large bundle of your DNA. Okay. So today we are going to look at how do these molecular scissors are used to understand the code of life. Okay, you can see in this picture, scientists actually editing the DNA and basically editing the DNA to modify something in the DNA so that it will be reflected in the life process itself. Okay. In particular, today I am going to talk about a story that won Nobel Prize in Chemistry in 2020, which is for the story of CRISPR-Cas9 system. Okay, this is the story that I am going to talk about today. Now, if you look at the DNA, you all know that DNA is the basic unit of life, not the cell, but basic unit of life is still a DNA. Now, if you look at the DNA organization, right, you have your genome, right? You all know your genome is, what is genome made up of? Sorry? What is genome made up of? Like, so there is an organization of DNA, okay? So you have your DNA, and DNA is assembled into a gene, okay? Gene is something that will code for certain proteins, and that gene will be assembled further into something called chromosomes. And those chromosomes, you all heard that chromosomes, there are, chromosomes from your parents, both from mother and father. So all the chromosomes come together and then forms a genome. So for a human, there is something called genome, which is divided into multiple layers. So it is divided into multiple layers, and if you want to edit your genome, because genome is something that will define how tall you will be, or what will be the color of your eyes, and what are the diseases that you are inheriting from your parents, or what are the other basic aspects of your life that you have, all are defined by your genome. Although your environmental factors play some minor role in that, but most of it is already predefined in your genome. So basically, if you can actually sequence your genome, you will know what kind of diseases are you prone to, approximately what will be your height that you can grow up to, or what will be the color of your eyes, what will the color of your hair, because for us Indians, the color is all black. But if you go to other Western countries, you have variation in the colors of the hair as well, right? So that will also be defined by your genome. So genome is something like, if you actually have to put an analogy, the genome is your storybook, right? Genome is your storybook that has all the information about your story. So your life is a story, then all that information is there in the genome. But then if you want to divide it, so you can divide genome into various hierarchies, right? So you have what is called chromosomes. So a storybook will have pages, each page will have paragraphs, paragraphs will have words, right? Words will have alphabets. So there is an entire hierarchy there. So the same hierarchy can also be observed here. So you have your story, and your story is in different paragraphs, which is your chromosomes. And then your paragraphs are nothing but your collection of your sentences, which is your genes. And then these sentences are nothing but they have a bunch of letters, which is your DNA. Yes.
Sir, aren't genes basically blocks of the DNA? So shouldn't genes make up the DNA?
So if you have a sentence, are you trying to say this? So sentence will have alphabets, right? Now there are alphabets in that. Now a meaningful sentence is there when the alphabets are arranged in a particular order. Now your DNA is these letters or these alphabets, and these alphabets are arranged so that it makes a meaningful sentence. So the basic, if you have a storybook, and if you say what is the basic unit of a storybook, it is your single alphabet, right? That alphabet is the one that is defining, it depends on the language, but it's still an alphabet that defines your storybook. Suppose you want to edit a storybook, you want to rewrite your story, you have to change the alphabets, right? Or that should make a sense, but what you are editing as such is your alphabet, right? Similarly, your genes are blocks which will code for proteins that have a functional role, but those genes are nothing but those genes are made up of DNA. So if you want to edit your gene, you have to edit your DNA. Okay. So this is the hierarchy that you have, where you have your DNA, gene, chromosomes, and genome, right? Now, when I am saying genome editing, in the sense, when I have to modify any of these parameters that define my life, if I have to edit a genome, at the end of the day, what should I edit? So just like an example I gave you, if you want to edit a storybook, you have to start editing the alphabets in that, right? So if you want to edit the genome, then you have to start editing the DNA of it. Okay, so for that we have to understand what is DNA and how do we edit that. What do we mean by editing a DNA? And so we have to understand what is DNA. Okay, I think still it's stuck. Yeah. Okay. So how do you think you can edit the DNA? What is DNA made up of? How do you know? How many of you know what is DNA? Deoxyribonucleic acid. Okay. What are the principal blocks in that. What is the DNA made up of? Sorry, say that again. From your group, yeah.
Adenine, guanine, cytosine, and
What are they?
they are
Bases, right? But does DNA have only bases?
A ribose sugar
A ribose sugar,
and a
yes,
phosphate group.
and a phosphate backbone, right? Now you have your, so this is your genome that I was talking about, and you can see the numbers here. The number of DNA in each of different organisms. For example, human has around 3 billion DNA base pairs in them, right? For example, a small bacteria will have around 4 million bases in that, or DNA bases in that. Okay, now coming back to this, so all this, as we say, so the function of your body is done by what is called as proteins. Now your proteins make up the basic functional forms of your body. So if I am talking, or my muscles are expanding, contracting, or my heart is expanding, contracting, so there are certain proteins that are around them that are doing this function. Now all these proteins are made up of your, basically this is called central dogma in molecular biology, where your DNA is converted into RNA and RNA is converted into proteins. So as you just said, DNA is made up of various bases, right? Now 3 bases code for 1 amino acid, and so basically you have some, somebody just said A, T, C, right, G or something like that. Now 3 of these bases code for 1 amino acid. Now when you have a entire bunch of A, T, C, G, A, G, U, A, G, T, A, C, something like this, right? You have a long chain of your DNA. Now this is converted into RNA, right? This is your DNA. DNA is converted into RNA. The only difference between RNA and DNA is if you look at the structure of DNA and RNA. So this is your DNA, which we were just talking about. You have a phosphate backbone, and then you have a ribose sugar. On the ribose sugar, you have 4 different bases, right? So these are your AGCT or ATGC, depending on how you want to call them. Now the difference between DNA and RNA, do you know what is the difference between DNA and RNA? Uracil is replacing your thymine, and what else? That is the structure of it, but I am talking about the chemical composition. Ribose sugar and deoxyribose sugar. So basically, if you want to look at this, so in your RNA, you have a hydroxy group here, whereas in your DNA, this hydroxy group is replaced by a deoxy, which is your hydrogen. So basically, you have your DNA and RNA. Now, going back to this central dogma, so you have DNA, and DNA is transcribed into RNA. So the only difference is that your T is replaced by U, A, U, C, G, A, G, U, A, C, something. And in this is your DNA, this is your RNA, and there is something called ribosomes that will code, basically they'll read 3 of these bases, okay, they'll start reading 3 of these bases, and each 3 of these bases will code for 1 amino acid. So individually these will start coding for 1 amino acid 1, amino acid 2, amino acid 3, something like that. So they start coding for amino acids. And once you have a chain of amino acids, that will fold and that will form what is called as protein. Okay, we'll talk about proteins more in my next lecture, but today we will stick to ourselves to understanding the DNA. Okay, so you have your DNA, DNA is transcribed into RNA, RNA is translated into protein. Now suppose these proteins are the workhorses of the cell. Workhorses in the sense, your function of your body is defined by the proteins, right? So suppose you want to modify the function of your body, or you want to modify the function of any living system. This is not just for human body, but any living system. If you want to modify the function of that, at the end of the day, what you have to modify? You have to modify the DNA. Once you modify the DNA, it gets propagated, and that can get changed in your protein level. Okay, so in today's topic, we will see how to modify the DNA. Okay, now as some of you know, so DNA is a double helix. So basically, why will it form a double helix? Why will it form a DNA? You said DNA forms a double helix, whereas RNA doesn't form a double helix, or RNA is a single stranded, whereas DNA is a double stranded. Why do you think it is double stranded DNA, whereas RNA is a single stranded? there is an oxygen missing, so it has to form a double stranded. That is not, or that's partially, I would say that is not correct. Can you guess? For a stability, what kind of stability? Hydrogen bonding. So you know what is hydrogen bonding, right? So if you look at the bases of these are all your bases, right? Adenine, guanine, and then so adenosine, guanine, cytosine, thymine. So they have these oxygen nitrogen centers, right? So and then you have, so this is one DNA, and then you will have another DNA. That DNA will have hydrogen. So when a hydrogen is between two electronegative atoms, it will try to bridge those two electronegative atoms, right? So you have an oxygen and nitrogen here. You have a nitrogen and nitrogen here, right? So this will try to bridge those, hydrogen bonding will try to bridge those electronegative atoms, and then that will give the shape of this double helix, which is called as DNA double helix. Whereas this is not possible in the RNA. Why is it not possible? Because the bases are exactly same. You just have a uracil instead of thymine, right? The bases are the same. So if the hydrogen bonding is something that is helping you to govern this double helix, then RNA should also form a double helix, right? Because the hydrogen bonding is being formed by the bases, and the bases are conserved except for T. But the problem here is, now you have what is called as OH group at the end of your RNA, right? So how many of you know what is called steric repulsion? You all know steric repulsion. When you have a bulky electronegative or electron-rich atom, electrons are of, if the charge on any system is same, they'll try to repel. You all know that the charge on any system is same, they'll try to repel. So you have OH here. Now if it tries to form a double helix here, the OH of this and OH of this they are too heavy, they have a high electronegative, basically they are electron-rich atoms, right? They'll try to repel each other and it will not be stable, it will fall apart. Whereas here you don't have OH, you only have a hydrogen, and the hydrogen and the OH, so there will be no repulsion. They'll nicely form this double helix and form this hydrogen bonding to minimize the energy. Whereas that minimization is not possible here because you have a huge repulsion coming from your OH group. So you are, at the end of the day, any structure that forms is because of energy minimization, right? Now that energy minimization is not possible in RNA, whereas in DNA it will be possible. So this forms a nice double helix and this doesn't form a double helix. So that is the major difference between DNA and RNA. Now coming back to DNA and RNA, so basically now from now on, what I am going to do is, I am going to represent DNA and RNA with something like this. This is like some kind of your railway tracks here, right? So because every time it is not possible for me to write the entire structure because I'll miss on the details. I'm trying to explain you something, but if I keep on concentrating on that, I'll miss the details. So I'll be writing it something like this. This is my double-stranded DNA. Yes.
Sir, hydrogen bonding in DNA is possible. Is it because of the character of the anti-parallel character, sir?
What 5 prime 3 prime you're talking about? So yeah, so if you go into details, yes. So it's basically a 5 prime 3 because if this is 5 prime, if this is also 5 prime to 3 prime, then your bases will be this side, the bases will be this side. Then there is no interaction. Your bases have to be this side. For this, the bases to be this side, your strand should be other way, 180-degree rotation. So this is your 5 prime to 3 prime, this is your 3 prime to 5 prime. So that is what actually happens there. Okay. So now what I am trying to say, basically represent you is, so you are going to have, I am going to represent your DNA and RNA by these railway tracks from here, where, can you concentrate here? Yes. So we have so I am going to represent these individual bases with the help of this alphabets. When I say A, that means this entire nucleotide, okay. So I am going to simplify my representation, structural representation of DNA by using these railway tracks, and for RNA it will be only single stranded. I told you, right, RNA can only exist in a single stranded DNA or single stranded form because of the repulsions, and DNA need not be only in double helix. There are many other forms, but for the simplicity of this lecture we will stick ourselves to a double helix part, and this is going to represent my DNA. So this is my DNA and this is my RNA. Now let us come to what is called as genome editing. So the topic is to understand, so where did we start? We started with the topic that I want to edit the code of the life. Now you understood what is the code of the life. Code of the life is DNA, because once you edit DNA, it gets propagated, and then that gets basically that will show up in the other life forms. Now how do I edit the how do I edit the code of the life? How do I edit the DNA? Now, editing in this sense, we are talking about editing means what? So this is not a movie editing, right? What are we talking about editing here? So we are talking about, so this is your chair, right? This is your DNA helix, or this is your DNA strands. When I am talking about editing, what I mean means I can actually cut these bases wherever I want, and I can put any other new base that I want. For example, as I told you, suppose you have this DNA. This is the DNA sequence I have, right? Now I don't want this amino acid here, but I want some A4 amino acid which will give me a different property in the protein. Now if I have to have an A4 amino acid, suppose instead of G, if I have A here, this will result in a different amino acid, right? Now if I have to do that, then what I should do, I should either change this to A, right? So that is basically your editing. That means I am removing a part of it and inserting a new part of it. Or what I could do, I could also, suppose you are aware of hereditary diseases, right? So hereditary diseases are caused by specific gene fragments that are there. Now suppose a simple example, although this is more technical, a simple example is there is something called CCR5 gene. Okay, let us not get into names. There is something called CCR5 gene. Now this is, so you all know what is HIV/AIDS, right? That's a disease, that's a deadly disease. Now a virus takes entry into your system and it will take over your entire immune system, right? The virus actually kind of enters into your system and takes the help of this gene called CCR5 gene, and if it takes help of this CCR5 gene, and then it propagates. Now suppose you want to have human population where they cannot be infected by HIV/AIDS, then what you can do, if suppose this CCR5 doesn't have any functional role, you can always delete this particular CCR5 gene from your body. So in the embryo itself, you can delete the CCR5 gene. So this virus cannot actually even enter into it, right? So you can do that editing part of it. That means you can delete a part of your gene, or you can change the bases, or you can add new things. So you can delete, you can add, or you can substitute. Here I have substituted. In this case, I am saying I can delete it, right? In the other case, I can add new bases. So all this is possible when you have all these forms what is called as gene editing. In a sense, you are basically doing these three processes of substitution, addition, and deletion of genes. Genes means your DNA bases. Now, how do I do it at a chemical level? Now, let's come back to the basic unit, because when we are talking about all this gene editing, all this, what is happening in our body is a bunch of chemical reactions, right? So all this is happening because of your bunch of chemical reactions. Now, when I am talking about DNA editing, so what I am talking about is basically a chemical reaction in which is done by enzymes called nucleases. So what they do is, this is your DNA strand, basically I have tilted it. Now you have a phosphate backbone, you have a ribose sugar, and then you have a base, right? Now you have enzymes that can selectively break this phosphodiester bond. So you see there's a PO bond there. So the enzymes can actually selectively break in the sense, I'm talking about hydrolysis reaction. So break in the sense, they are not going to literally break anything. What they are going to do is, they are going to do a hydrolysis reaction so that this PO bond breaks and it gives two fragments. So in that case, basically what I am doing is I am removing a part of it, and so this is the chemical reaction that I am talking about when I am talking about gene editing. Gene editing, or so this is done by specific enzymes called nucleases. Now, how many of you know what are enzymes? Most of you know enzymes. What are enzymes? So these are biomolecular catalysts that speed up the reaction. Now there is a nomenclature for all the enzymes. When you see nucleases, so anything that has ases means that does a hydrolysis reaction. Okay, ases means something that does hydrolysis reaction, and nucleases means it does hydrolysis reactions on the nucleotides. So this is your nucleotide. So that will do a hydrolysis reaction on the nucleotide, and this will break this phosphodiester bond, and it will give you two different fragments. Okay. Now these nucleases can be of two types. Okay, one is called as endonucleases, other is called exonucleases. So what do you think is the difference between endonucleases and exonucleases? Okay, that is from the endothermic reaction. Endothermic reaction absorbs, exothermic reaction gives out. But so nucleases are not going to absorb any energy, right? So exo is a general Latin term that is used for endo means inside, exo means outside. Okay, exoskeleton, endoskeleton. You must have, so some organisms have exoskeleton. For example, if you look at a tortoise, the tortoise skeleton is outside, right? That's an exoskeleton. Whereas we humans have our skeleton inside. We are endoskeletal species. So endo means from inside, exo means outside. So there are these different kinds of, two different kinds of enzymes that do this hydrolysis reaction. So we are talking about editing the gene, and the editing the gene means basically doing the hydrolysis reaction, or basically breaking the phosphodiester bond. So the enzymes can do both exonucleases, which will cut out the phosphodiester bond from the termini, or endonucleases, which will cut out the phosphodiester bond from the inside part. So which one of you think, which one of these two you think is more important or interesting? Endonucleases or exonucleases? Why do you think? You are right that endonucleases are more important. Why do you think?
Because if we cleave DNA from the inside, we can access specific fragments at a time.
Very good. So now you have a DNA. If I have an exonuclease, I have to keep on doing this. Suppose this is the gene I want to edit. Right? This is the DNA I want to edit. Now if I have to use a DNA exonucleases, I have to cut all of this to even cut this. Whereas if I have endonucleases, that can specifically break any of these internal bases, that will be more useful, because that will help you to have a grip on modifying anything that you want. Whereas here you are only restricted to termini. Whereas if you have an endonuclease, you can start editing something from the middle of it, because your DNA, as I showed you, these are very, very long chains, right? So if you start doing it from the end, it will take probably eternity for that to come to the middle, right? And you lose all the information that is there. If you want to only specifically target few regions, it is always better to start doing it from the middle of it, or the region of our interest, and that's why endonucleases are very, very important. And I was looking at the NCERT textbook, and you have a introduction to endonucleases in your 12th class biology text chapter on biotechnology. So this will also help you in that, but I'm going to go beyond that, because I don't want to teach you what you will be learning in your 12th class, but I'm going to go beyond that. But because you don't have this basic understanding, so I'm trying to tell you that there are two different nucleases, endo and exo. Okay. Now, yes.
So, how do you know where the endonuclease is going to cut the gene?
Very good question. That is what we are coming to. Very nice. Okay. Now, this endonucleases that I said are enzymes. Nucleases are enzymes, right? There are variety of enzymes, and their discovery is not something that I'm going to talk for now. But there are variety of enzymes. And how did people actually realize that? We are going to see that in the other case, but people have developed, or people have understood. So this is an enzyme structure, okay? Some enzyme structure that is binding to a DNA target. Okay, we will talk about enzyme structures, but just for now, assume that this is an enzyme that is binding to a DNA. Every enzyme has a specific sequence, just like how we asked. So every enzyme has a specificity in it, in the sense, an enzyme, for example, this is a red region that it wants to break, right? Now you have an enzyme. I'm representing this enzyme by two ovals here. Okay, so this enzyme can bind to this nucleotide. You have a large nucleotide, and it can bind to this nucleotide, and then it could either do a non-specific binding, and then it can do a diffusion. And once it knows the region, because every enzyme has a specificity. Most of the enzymes work by what is called as lock and key mechanism. Okay, where you have an enzyme is a lock, and there is a specific key that it binds to, right? So basically, you have an enzyme that can diffuse on the linear strand of DNA, and once it finds its key, it can actually bind to that. Or you can also have a specific binding, where you have your enzyme and you have your DNA, and the enzyme can bind specifically to a specific part of DNA, and then it will do the hydrolysis reaction that I just showed you. And this story of how these restriction enzymes, these are all called restriction enzymes or endonucleases. The story of how these restriction enzymes were discovered itself is pretty interesting. But when I talk about CRISPR a bit about it, so we will talk about it. But just for you to understand, so if you look at this, so these are different enzymes, okay? These are different endonucleases or restriction site. enzymes. So you see every enzyme has a specific restriction. For example, there is an enzyme called BAMH1. Okay, that is specifically recognizes between GG, guanine, when there are two guanines. So each of them can actually recognize a specific base and it will start cleaving it. Okay, now if you see this particular thing, this restriction enzymes have a peculiarity in the sense they only recognize what is called as palindromic sequence. You know what is palindromic sequence? Right? So something that you read from the front would also, that will also be, you will be able to read exactly the same from the back, right? For the most common example word is what? Can you tell me of a common example of a palindrome? Race car. Okay. Yeah, so you know what are palindromes. So if you see this, all these are palindromic sequence in the sense GGA TCC, then it will be GGA TCC. Just like somebody asked, there is a parallel and anti-parallel strand. So this is your parallel strand, anti-parallel strand. GGA TCC, GGA TCC. So for most of the restriction enzymes, don't go to this here, but for most of the restriction enzymes, these sequences are actually palindromic in nature. So if you look at these things, so they're all names of the enzyme. The way these enzymes were discovered itself is pretty interesting in the sense. So how are they discovered is basically now, how they were discussed is you have a bacteria, right? So a bacteria is attacked by a virus. Okay, that's a bacteria can be attacked by a virus. So this is a bacteriophage. Okay, once this bacteria basically has seen this virus, the virus will incorporate its genetic material, either it could be DNA or RNA into this, right? Now when there is a lot of this genetic material from the virus in this, the bacteria actually it's a part of this immune system where the bacteria will recognize a specific DNA sequence and it will start cleaving into it. So this entire process of this endonucleases are part of DNA, are part of bacterial immune system in the sense these were not discovered or these were not invented or discovered for our use. They are actually part of the bacterial immune system. Okay, so we will as I said, this is something that you learn in your 12th class, so I'm not going to detail about it, but I'm going to talk to you about the part that you will not learn in 12th class, which is something that is beyond this.
Sir,
okay, yes.
What is N?
N means any nucleotide. So you have a specific nucleotide and then you have any nucleotide. So it could be any nucleotide, yes.
You said that the enzymes will, recognize the particular part and then the cleaving starts happening. How exactly does it recognize that?
That is a very, very complex process. So how enzymes work is not, so we will, as I told you, so enzymes are made up of what? Proteins. Proteins are nothing but amino acids, right? So every amino acid has a specific chemical character, like there is a lysine, there is a NH2, right? And there is an aspartic acid. So these amino acids have specific chemical behavior in that. So that chemical behavior will have a chemical interaction with one of these bases specifically. So although as I told you, we are just showing it as a cartoon because they are very, very complex structures, right? So that specificity comes because there is a difference in the chemical nature of every enzyme, and difference in the chemical nature will actually move to difference in the specificity of the binding. Yes.
Sir, you said that each of these enzymes, they target their own portion of the DNA which they bind to. Is it possible for two of these enzymes to be combined so that that resultant enzyme gets the properties of both of them? So it goes for like for example the first one and the second one's properties, like that.
Yes, so that's a good question. So there have been studies where people have actually combine two different enzymes, but the problem with that is all the enzymes are, so you have a binding that actually goes into it and it cleaves it. Now it cleaves it, so basically the second enzyme can only act because your two sites may not be close by. If they are close by, then that would make sense. If the two sites are far apart, then this has to do its action and then the other one has to go and do its action, right? Instead of that, you add both the enzymes separately, independently, then both of them will do their action separately, right?
Sir, what if there are multiple locations where the enzyme can go? What if there are multiple GGs but you want to attack only one? You don't want to attack the other.
So basically for an enzyme to recognize, so it needs to have all these 6 bases, right? For example, an enzyme called BAMH1. So if this enzyme called BAMH1 can recognize this GGA TCC, if all these 6 bases are there, only then the enzyme will recognize the nucleotide, but it will do the cleavage. Cleavage in the sense it will actually break the phosphodiester bond at this. Whereas this enzyme will recognize only if all the 6 bases are there. So for recognition, it requires all the 6 bases, but for breaking the phosphodiester bond, it will break at a specific location. For example, between the A and A here, or A and A here. Here it is between G and A and G and A. Okay. So this is something known as endonucleases, where you have a specific gene or there are specific bases where the enzyme will go and bind to it and break it. Now, as I just told you, let me complete. As I just told you, you have a human genome which is it has few billions of bases, right? Now, it is almost impossible to have a particular gene or particular enzyme to go and attack different parts of it, right? There are 4 bases, and there are, if the number of bases are 3 billion or 3.2 billion, then you have a various combination of these 4 bases. So to have an enzyme for every specific combination, it is almost impossible. Can somebody guess what will be the number of combinations that are possible if you have a 3.2 billion genome length that is just made up of 4 bases? You have only 4 bases. 3 billion to the power? What will be the number of combinations that are possible? There are 4 bases. So there are many combinations that are possible, and it is impossible to have all these enzymes, so many number of enzymes that can target individual species, right? Now suppose suppose my restriction enzyme is there that can only target this. Now it is not possible for me to target this with that enzyme. Then I have to design a separate enzyme. So that way you will have a many number of combinations, and it is not possible to have so many number of restriction enzymes. So restriction enzymes are only limited, and they will only, for example, I have given you few examples. So they can only look into this particular few bases, and they will go and specifically actually cleave them. Yes, what is the question there?
So the last enzyme in the list, it's written as non-specific. So how do you use that for?
Good question. That is what I am coming to now. So these are all specific enzymes that actually go and bind to a specific basis, and then you can actually cleave that. Now there is something called non-specific enzymes. Non-specific enzyme means it will cleave any DNA that is there. That is very dangerous, right? Because now if you are not giving specificity to that, you give it to a cell, it will start cleaving everything. So you need to have a specificity to that. But how do you add specificity is something that I am going to tell. Yes. Yeah, tell me. The slide is gone. Tell me.
So in that, it was given 9n, I think. 9n, it was cleaving. 9nn. What does that mean?
9n9 means
there was
nine different nucleotides.
Okay.
Just any nucleotide it could be.
Any like specific conditions for these enzymes to work? Like
yes. So basically, most of the enzymatic reactions that we talk about occur in a normal physiological conditions. When I say physiological conditions, I am talking about somewhere between 30 to 40 degrees centigrade, and then you have a pH, neutral pH close to. So enzymatic activity is more active. Enzymes are more active in the physiological conditions because these are biological molecules. But there are enzymes that are active under, for example, there are many living organisms that live under extreme conditions. There are organisms that live in hundreds of geysers. You know geysers, right? Hot springs. There are bacteria in that geysers as well, right? So you need to have specific enzymes that are also active in that. So there are enzymes that work at that temperature, but most of the enzymes are at the physiological conditions that we are looking at.
Sir, would the body have like a repair mechanism where it will notice that the DNA has been cut somewhere, like a kind of immune system? The DNA has been damaged somewhere and it goes and repairs it and undoes our changes. Is there
Sorry, could you repeat that?
Sir, we are editing the DNA now, sir. So would the body have a kind of response
repair
reaction
mechanism.
to that repair mechanism?
Yes,
Mechanism which will undo our changes.
Yes, it has a repair mechanism. That is called as ligation. In the sense, basically, when something is broken, you are using this enzyme to break the bond, right? Nucleases. But then if it has been done by mistake, there are enzymes that look for these, and then there are enzymes that can make this phosphodiester bond, which are called as ligases. So you can break the phosphodiester bond, which is the hydrolysis reaction. In the other case, you are basically forming this PO bond. Those are called as ligases, which form the PO bond. Okay. Yes.
Would we have any uses for exonucleases?
Exonucleases are more useful when you are actually doing something called as sequencing, like when you have to do a sequential digestion of something, but the uses are very, very limited. But there are uses where, for example, now as I just told you, there are phosphodiester bonds, right, that forms. Now if suppose that phosphate is not, like there is not enough phosphate and you want to go to a next base, then you have these exonucleases that will break that phosphodiester bond so that the next base can form the ligation mixture. Okay, so there are uses, but there are not as many uses as your endonucleases would have. Okay, so now let's move on to, let's move on to this and then I'll come back to guys. Okay, now the question that because I didn't even get into what I wanted to tell you. Okay, so what I wanted to tell you is, so this is something that is known. These are something called endonucleases. As I told you, if you are an NCERT textbook, you will have some information on endonucleases in the 12th class. But there are something called endonucleases that specifically recognize a basis and then you'll do it. But then, as I told you, it is impossible to have so many enzymes for all the possible nucleotide sequences, right? So you have many zillions, I would say 4 power 3.2 billion number of combinations that are possible. So it is not possible. This is just humans I'm talking about. But there are other organisms, right? So it is not possible to have so many enzymes to specifically recognize every possible, if I want to break something where I want, that is not possible. You are only restricted to where the enzyme can bind, only that part you can break. But suppose where I want to bind, that I cannot do, right? So there you can use what is called as non-specific endonucleases. But the thing is, you need to have a specific tag, or you need to have something called guide molecule that will bind, that will go and bind to whatever region that you have. Now here the enzyme can only recognize this specific thing in the specific endonucleases. But suppose if I want to break or if I want to modify anything that I want, then I need to have a non-specific endonuclease, but that should be guided by a guide molecule, and that can take you to anywhere that I want to edit that. So that is what this particular system has done. This is called as CRISPR-Cas9 system. This was awarded Nobel Prize in Chemistry in the year 2020 for developing a system where you could edit the genes at any place you want. Okay, so we are going to talk about that system, but before that, somebody had questions here.
So what if the ligation genes are self-cleaved?
Sorry, say that again.
The genes for ligation, the enzymes, like there are enzymes which are made from the genes, right? So what if those genes are cleaved?
How are those genes cleaved?
No, what if those genes are cleaved?
I didn't understand the question. Can you repeat that?
So like you said that nucleases,
ligases are again enzymes.
okay, yeah.
Right? These enzymes are made up of your proteins. Now these enzymes can go and make those ligation bonds. So just like how we had endonucleases that break the bond, there are ligases which can make the bond. So these are two different kinds of complementary enzymes.
No, so I was asking that ligases are made from certain genes, right? So what if those genes are itself cleaved?
So okay, now so basically, okay, so because I have skipped few things, so now this entire cleaving part, this is done, what did I say? This is done as a mechanism for defense. These were originally made for defense mechanism. So basically when you have a bacteria, right, now when it, I'll probably tell you that. Let me skip few slides. Okay, now you have a bacteria that is infected by the virus, right? Now that virus will inject a specific DNA that it wants. Now over the years, over the evolution, the bacteria has known, or bacteria has developed enzymes that can recognize the bases from the viral DNA, and then they will specifically go and cleave that. But the important question is, how will it prevent its own DNA from cleaving? Because it is going and cleaving the viral DNA, but it has to prevent its own DNA. It should not have its own DNA cleaving, right? For that, there is a specific mechanism that has been developed that is called methylation, where you have your DNA that will get methylated, and that will prevent. So you have your DNA that has been methylated, and that will actually prevent it. But that's a different topic altogether. So you can have your bases that can be methylated, and those will prevent it, but that's a different, that is not a part of the story that we are talking about. So but the bacteria can recognize its own genes and prevent them from degradation or cleavage, whereas it can recognize the viral genes and can specifically do the cleavage part of them. Yes.
Sir, when we program, like when we use guided nucleolysis, how does it, like when there are two same bases, how does it distinguish between them?
Good question.
Like
So
one
that is,
might be on the fourth thing, or and another might be on sixth one. I want to remove the sixth one. How do I make sure it does not remove the fourth one?
so okay. Now, so basically you have your DNA, right? Now we talked about complementarity. Now suppose I have this, right? Now I have this DNA ATGCAGTACTAG, something like this, right? Now I don't have any endonuclease, but I want to remove this particular part, or I want to remove this particular region. Now we just saw that. So there is a complementarity. Always A will bind to T and G will bind to C. Do you know what is Chargaff's rule? Right? The amount of ATGC content is constant. So A will always form a hydrogen bond with T and G will always form a hydrogen bond with C. So if you want a specificity, suppose you have a CGGT, then you need to design your guide molecule that will have what? G, T, C, A, right? And then you have your non-specific enzyme. So this will only go and bind here. This will not bind anywhere else because there is no complementarity. So this will have a specific complementarity because A can only bind to T, G can only bind to C because of the number of hydrogen bonds. So this will only go here, and then there is a specificity that will come. Once this specificity is established, then you can define the length of this particular thing so that the enzyme can actually break. Suppose you want to break it here, then you have to design your guide molecule somewhere farther based on the length of this and how far your enzyme can bind. Yes.
Sir, here the part of the DNA we want to remove is the unique part in the whole sequence but if there's a repetition of the part which we want to remove, then how do you
so
which one
that's a good question. Now repetition is only for a specific region. For example, I have this CAGT repeating multiple times, okay, but I do not have, repetition will stop somewhere, right? Now CAGT AC, but there will be CAGT GT probably. So you'll also include this part of that, and then you increase the length of this in such a way that it will only bind to this specific region and this will only cleave it somewhere here. So there is a repetition, then you need to come out, because there are only 4 bases, right? So there is a good chance that there is a repetition. So you have to make sure that you are actually coming out of that repetition so that you can actually have a DNA guide molecule that will bind to specific location that you want.
Then after that we'll add the things we removed extra because we didn't want that. Like you told that CAGT will repeat, so we'll take CAGT AC which does not repeat, let us assume.
Right.
So we'll again add back AC.
We can add back those 2 bases that way. Yes. Okay. So yeah. So specific endonucleases are something that I told you, whereas what would these molecules do is to look at this guide molecule and they'll do what is called as non-specific degradation of that, and the system that has been developed is called as CRISPR-Cas9, and as I said, that has been awarded Nobel Prize in the year 2020 for chemistry. And there are few things, I guess all of you know what is Nobel Prize, right? So it is the highest honor that a scientist could get in terms of their scientific discovery. Okay, so this is a very novel discovery that happened. So this all started in the year 2011 or 2012 and it won Nobel Prize in the year 2020. Generally, Nobel Prizes are not awarded for such a short, you have to do a research for a really long time to establish a concept. But this particular concept of genome editing has become so popular that it won Nobel Prize within 9 years of its discovery. Okay, and so this has been awarded to 2 female scientists, Emmanuelle Charpentier and Jennifer Doudna. It is important to note that this is the first time that 2 women has shared a chemistry Nobel Prize. This is the first time that ever happened in the chemistry that 2 women have shared a Nobel Prize. So this Nobel Prize itself is pretty interesting in the terms of both the number of years it took to reach to that level as well as the people that got it, and one is from UC Berkeley, the other is from Institut Paris. Okay, she did her PhD in this. So These are the people who got Nobel Prize related to this. But before I go into this, I wanted to tell you about how this system works, because I didn't even come to the part where the how this system, we are only giving a prelogue to this. Yes, you have a question. You had a question? No? Okay. Okay. Now, let us understand the history of this particular CRISPR-Cas9. Okay, how was it discovered? So, in the year 1987, so basically long back, somebody have observed that few bacteria has this peculiar gene. I told you, right, I am going to represent all my DNA like this. They had this peculiar gene where they had this palindromic repeats, and so these black ones are the palindromic repeats. And in between these palindromic repeats, there are some other foreign DNA that is there. So this was the discovery. So most of the science initially happens through this discovery. So people have isolated DNA from certain bacteria, and they have observed that it is very surprisingly there are these specific repeats, palindromic repeats, and then there are these non-specific foreign DNA that is placed in between these particular palindromic repeats. And it was named as CRISPR. CRISPR stands for clustered, regularly interspaced, short palindromic repeats. That means these are clustered, these are in a cluster, these are short palindromic repeats that are interspersed by this foreign DNA. There are other DNA that is there in between these palindromic repeats. So this is what was observed during a specific experiment. Yes.
Sir, when genome editing is done, how will we identify which amino acid codes for this particular physical characteristic?
So, okay, that again has to go back. So, you know which code will code for which amino acid, right? Now, this comes from, so this is not a single day study, right? This is a study that happens over decades. You first identify the proteins that are responsible. For example, now I am seeing a color of my eye as blue, suppose. You will identify which protein will actually give me that color, right? Or you are doing some, you are having or some diseases there. You will identify which protein is responsible for that. And then once the protein is established, and once you have a human genome, you can do a genome sequencing. You can sequence the entire DNA that you have. So you all probably know that you can do it. So you can give your DNA and you can get it sequenced. And it is very, very common in abroad, but even in India it is catching up, where you can sequence your entire DNA. And then, so the protein that is responsible, so you do mutations in the sense, there are specific binding region where it binds. So you replace those few amino acids and see which one is causing that particular disease. And you identify the amino acid. Once you identify the amino acid, you map back to see which part of the gene is coding for that amino acid, and then you do. So it's not like it is not front to back, it is always back to front. You first identify the phenotype, that means what is causing that, and then from that you determine what protein to amino acid to base. You always come from the back. That's why study in biological research takes few decades, right? It takes few decades to identify one small thing. Yes.
So I can say that an amino acid causes some sort of trait in a person.
Not one amino acid. There could be a bunch of amino acids.
Can I say protein causes it, like one?
Yes. Yes.
Okay. So when we're using a nucleus, a non-specific type, when we're using a guide molecule, when we're targeting it and the amino acids are repeating, does it target all of them that in that strain where you?
So I'll show you what is called. So if you all have internet, you can go back and see what is called codon table. Nature is pretty impressive, or nature is pretty intelligent as well. So you have a codon table. One amino acid can be coded by 6 different bases. That means, so ATG, or suppose CAG, right? Suppose it codes for A1 amino acid. The same thing can also be coded by TAG or GAG, something. So a same amino acid can be coded by 6 or more, or sometimes 6, sometimes 2, sometimes 3 different bases. So when you have different amino acids in there, it is not necessary that all of them will have same bases. They could be coded by different bases, which will cause to this. So I don't know if you have, if we have internet here, I can show what is, or maybe in the next class I'll show what is called codon table, where every amino acid can be coded by multiple bases, and you could have the same amino acid but different gene sequence. There is a possibility. But suppose you don't have that possibility, and also the repetitive nature of these amino acids is pretty less. So if you don't have that, then you have to come up with a different strategy to look into it. Okay, yes.
So
Just a minute. I think I have to ask the time. I'm scheduled till 10, right? Scheduled till 10, right? Yeah, tell me.
Sir, you said that each amino acid has 6 different representations.
I'm not, just 6 is a number. You could have just 1, 2, 3. It could be, still maximum is 6.
Maximum is 6.
Yes. Maximum is 6. You could, for example, something, an amino acid sometimes could be coded by only one codon. That means 3 bases.
So is that based on how common that amino is?
That is also one of the hypotheses, but this is, in biology, it is more of a nature driven. This is what is observed, and why there is a logical thing is something that is, we only hypothesize that. That is something that you can say that based on the how common that amino acid is, there could be a number of bases. Okay, yes.
Sir, CRISPR, like there is palindromic in the name for CRISPR, right? So is it only used for palindromic sequences or?
No. So basically this, when this was observed, as I said, this black one that we are seeing, right? This is a palindromic sequence, right? So there is a palindromic, sequence which is interspersed by some other foreign DNA. So that is why it is called, so this CRISPR technology that we are going to talk about will have only palindromic repeats with these non-bacterial DNA. That means some kind of foreign DNA it will have. Okay. Now I think I will not have time to go into CRISPR. This I will leave it for my next class, which is on Wednesday. Okay. But this is something I will also show you the codon table, like how different amino acids are, coded by different bases. Okay. I'll also talk about that. Okay. Yes.
Sir, could you briefly explain how the bases are represented by those boxes that represent?
So this is exactly what I have it here, right? So each of those boxes, right? There is A, there is a complementary. A is complementary to 2, then G is complementary to C, G is complementary to, so each of this will have, this is one base and this will be its complementary base. This is one base, this is its complementary base. This is one base, this is complementary base. It could be any sequence. I am taking a general case, but it could be any sequence there. But the top one will suppose is A, the bottom will be T. The top one is G, the bottom will be C. So that is the chain of, so each of those lines, the top is one base, the bottom is one base. Okay. Okay, any further questions? Okay, if not, then let's stop it here, and then we will have the next class on Wednesday, where I'll try to complete this CRISPR part.